Good afternoon, everyone, and welcome to today's Biomarkers series webinar, High Throughput Biomarker Discovery for Clinical Trial Optimization. My name is Esther, and on behalf of Oxford Global and Sapient, I would like to thank you all for joining us today. After the presentation is complete, we will have a 10-minute Q&A session. Please submit your questions through the question panel on your menu display. The full recording of this webinar will be made available to download on our secured website, and the details will be sent to you shortly after today's session. Advances in mass spectrometry resolution and sensitivity have enabled increasingly comprehensive views into the multitude of small molecule metabolites circulating in humans. These metabolites read out diverse hosts and environmental exposures that influence human health and may serve as critical biomarkers for driving early disease diagnostics, precision medicine approaches, and clinical trial optimization. In this webinar, Dr. Mo Jain will share how Sapient has established discovery as a service approaches that utilize next-generation high-throughput mass spectrometry, advanced data analysis, and population-scale data assets across more than 100,000 individuals to enable robust discovery of biomarkers that can be used to align patients, disease biology, and therapeutic interventions for clinical trials. You will learn about the role of small molecule biomarkers in providing insight into human disease biology and drug response, technologies enabling rapid identification of small molecule biomarkers of biological processes, disease progression, and drug response, including for clinical trial optimization, real-world examples of how Sapient's small molecule biomarker discovery approaches have been used to advance clinical trials. Dr. Jain is a physician scientist with more than 20 years of expertise in physiology, biomedicine, engineering, computational biology, and mass spectrometry-based metabolomics. Prior to founding Sapient, he formed and was director of Jain Laboratory at the University of California in San Diego. There, he led a multidisciplinary research team to develop next-generation rapid liquid chromatography mass spectrometry systems to probe the non-genetic landscape of disease across population-scale human studies supported by over 30 million in federal, foundation, and industry funding. Dr. Jain founded Sapient in 2021 as a spin-out of Jain Laboratory to expand upon the mission of accelerating human discovery and drug development through the nexus of high-throughput analytical mass spectrometry, computational biology, and population-level biomarker profiling. Dr. Jain has held faculty positions at UCSD since 2013, most recently as a professor of medicine and pharmacology. He obtained his MD and PhD from Boston University School of Medicine and performed clinical residency and fellowship training in internal medicine, cardiology, and preventative cardiology at Brigham and Women's Hospital, Harvard Medical School. So without further ado, I hope you all enjoy the webinar, and over to you now.
Thank you so much for the kind introduction, and we're very excited to be here today to present some of our ongoing efforts. I'm just going to go ahead and share my screen here. So as mentioned, we'll be chatting a little bit today about our efforts in biomarker discovery and how we leverage high-throughput bioanalytical tools, specifically for biomarkers within the context of clinical trial optimization. Now, the fundamental problem that we're faced with here today is the same one that many of you have spent many years working on, and this is the fundamental challenge with drug discovery. As many of you are aware, drug discovery is a challenge that's fraught, unfortunately, with failure, and there's various statistics around this, but certainly the majority of drug candidates that enter into lead optimization in preclinical studies will never see the light of day. And 1 out of 10 drugs that ultimately enter into clinical studies will ever gain regulatory approval and help patients. Now, this is really quite the challenge when we're dealing with over 90% of agents that ultimately fail through the clinical trial program. Now, when we subset this into where specific failures occur, when we look at early clinical trials, those in the phase 1, phase 2 area, approximately 70% of agents that are entering into this stage ultimately end up failing and not advancing. And arguably, this would be expected, as these are thought to be early clinical trials that really should be evaluating not only safety, but early efficacy. What's really challenging that is if we look at the terminal stages, the phase 3 clinical trials, which ultimately end up taking an enormous amount of time and expense, 50% of these phase 3 clinical trials also ultimately end up failing. Now, if we look further into why these phase 3 clinical trials ultimately fail, approximately 2/3 of phase 3 clinical trials ultimately end up failing for efficacy. So not safety, but rather efficacy, meaning at this terminal stage, the drug is not found to reach the primary endpoint. Now, oftentimes, this is not actually due to the drug itself, but rather challenges related to several different factors, including a heterogeneous disease and patient population, in which the disease itself may be quite heterogeneous, and as such, there may be variable response. In addition, there can be quite a bit of challenges with clinical trial optimization and clinical trial design, both under targeting of responder populations and then also ensuring that there's adequate outcome numbers to power clinical studies. Ultimately, this is all resulting in poorly ineffective translation, and this can also be due to insufficient understanding of PK/PD parameters, inadequate measure of target engagement, and many other parameters. Fundamentally, as sort of mentioned, what this results in is over 50% of agents entering into this phase 3 stage failing, and again, 2/3 of them failing for efficacy reasons. Fundamentally, it's not likely due to the drug itself, but rather challenges with the clinical trial structure, both due to heterogeneous disease patient populations, clinical trial design, and effective translation of the underlying agent. Now, even if we look among FDA-approved drugs, what we find is that there's great variability in underlying response rate. So if we look at over 12.5 targeted oncology therapies that have been approved in the last 20 years by the FDA, what we find is even among these agents, which are listed here on the right, that there's a median response rate of under 50%. Now, for those agents that are tested in multiple disease populations and multiple tumor types, what we find is that the more heterogeneous the disease, the more the actual response rate declines. And this necessitates larger patient populations, as you can imagine, as well as challenges in deploying these agents in an effective manner to ensure maximal efficacy while limiting any potential adverse events. Now, the reason for this has to do with a fundamental mismatch in how we develop and deploy drugs. So when we think about sort of drug development and disease diagnosis is really based upon an end pathology, whereby we're looking at a tissue specimen and a diagnosis is being made, and we have a host of individuals with a shared end pathology that all share this same diagnosis. Now, as we know, pharmacologic therapies, again, whether they be small molecules, large molecules, gene cell therapies, are targeting a mechanism by which an individual goes from a normal to an abnormal state. Now, everyone that has a shared diagnosis is fundamentally receiving the same therapy. And in doing so, what we find is that there's a portion of individuals that benefit, there's a portion of individuals that have no benefit, and there's unfortunately a portion of individuals that actually end up having an adverse event. And this mismatch between how we diagnose individuals, again, based upon an end pathology, and how we develop drugs that are targeting a mechanism from which individuals go from a normal to an abnormal state, is what causes this misalignment and ultimately drives some of the drug development challenges. And this is where biomarker-guided drug development can have profound influence on improving the overall efficacy of this drug development pipeline. Fundamentally, what biomarkers allow us to do is to take individuals, all of whom have a shared diagnosis, and subset these patients, and in essence, align individuals with their specific disease pathobiology. And in understanding their specific disease pathobiology, we can best align individuals then with the ideal therapy. And in doing so, we can optimize individuals, their disease pathobiology, and their specific therapy in a manner in which allows for maximal benefit across very diverse populations, again, all of whom share an underlying diagnosis. Now, this is more than just theoretical, and when we look in practice, this certainly has been borne out. And this is one of my favorite analyses that came from Albert Lowe, an analysis of over the last 20 to 30 years, over 20,000 clinical trials that span many, many therapeutic areas, from oncology to cardiovascular disease, autoimmune disease, vaccine development, et cetera, et cetera. And in this analysis, they really looked at all those factors that are predictive fundamentally of FDA approval, meaning the success of a drug moving through the clinical development pipeline. And what they found is the #1 predictor of whether or not a drug would ultimately be approved was whether or not that drug was developed together with a biomarker. Now, if we look across various areas such as oncology and cardiovascular disease, we see those agents that are developed together with a biomarker versus those that are developed without a biomarker have almost a 9-fold increase in ultimate FDA approval. When we look across all therapeutic areas in over 20,000 clinical trials, we find that as a whole, there's a 2x increase in approval rates when drugs are developed together with a biomarker. Now, not only is the approval rate higher, but the time to approval is faster. And those precision medications that are developed together with biomarkers are approved almost 20% faster than those agents that are not. Now, it's not a mystery that these sorts of numbers, being able to improve overall FDA approval as well as the time to approval, can be transformative for underlying drug development. Because of these findings, you can imagine the FDA has really embraced this strategy of biomarker-guided enrichment for clinical development. And these biomarkers can take on a variety of flavors. In a recent white report or white paper from the FDA highlighting the value of biomarkers for enrichment strategies, there's 3 main areas that we can think of these biomarkers as falling into. Biomarkers that denote individuals with a specific pathophysiology, once again aligning patients with their specific diseases. Biomarkers that predict response, ultimately allowing for alignment between a particular therapy and a particular disease state. And those biomarkers that speak to a specific mechanism of action that allow us to understand target engagement. Now, when we think about diagnosis of disease, certainly this is an area that biomarkers have had profound influence over the last several decades, particularly in the oncology space. And this one slide provides a really nice overview of just one such example. If we think about lung cancer, the traditional view of lung cancer just several decades ago was one that was, again, based in pathology, in which lung cancer was diagnosed based upon its underlying appearance on pathologic examination and was fundamentally divided into adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. Over the ensuing decades, however, as molecular biomarkers, particularly in the genetic space, have evolved, this has allowed us now to have much greater granularity and subset lung cancer into their underlying biomarkers that allow us to understand the pathobiology of the disease. Initially with KRAS, with EGFR, and then when we look at today, lung cancer now has evolved from a disease of 3 fundamental areas to a disease that's been subsetted into dozens of areas based upon these biomarkers, once again, that denote their underlying pathogenic mutations and that have profound influence in guiding fundamental therapy for this disease state. Now, the challenge is, how do we employ the same strategy of utilizing biomarkers for early diagnostics, for substratifying patients, and for clinical trial strategies? And how do we do this outside of the oncology space? And this has really been quite the challenge. Next generation sequencing, of course, has really enabled biomarker strategies within the oncology space, but outside of oncology, whether it be in the heart disease space, whether it be in the neurologic disease, lung disease, liver disease, et cetera, this has really been quite slowed. And in thinking of beyond genetics, it's important that we develop additional data modalities that allow us to understand these other disease states, particularly the internal and external exposures which influence biological processes. Now, there's many ways in which one can begin to model the influence of genetics on human disease, and this is just one way that we do so in thinking through large populations of mono versus dizygotic twins. And when we begin to map this, what we find is that the vast majority of population attributable risk for all these diseases, again, whether it be autoimmune disease, cardiovascular disease, neurologic disease, et cetera, lies outside of genetic influence. And the genome only influences approximately 20 to 25% of disease risk. It's important as we think about how to step beyond genetics and develop biomarker strategies for these other disease states that we're able to leverage these non-genetic biomarkers, particularly for drug development and deployment. And this is where Sapient has spent many years thinking through our strategies. The areas that we've particularly been focused on are what we call small molecule biomarkers. Now, these are biomarkers that represent both endogenous as well as exogenous markers that are floating around in our body. These typically are small molecules, as denoted by the name, and have a molecular weight that lies below 2,000 Dalton. And when we think about these small molecule biomarkers, or metabolites as they're oftentimes referred to, these can complement traditional genetic biomarkers and allow us to read out multiple determinants not only of disease states, but also drug response. Now, what makes these small molecule biomarkers particularly unique is that they can be produced intracellularly, both in normal tissue, in diseased tissue, within the microenvironment, or even in exogenous areas. And given their physical chemical properties, these small molecules can readily cross biological membranes, whether they be cellular membranes, the blood-brain barrier, et cetera, which allows them to move from the intracellular state into blood, where we can measure these non-invasively as reflections of what may be occurring in the tissue, again, whether it be the normal tissue, the diseased tissue, or even the microenvironment. Now, these small molecule biomarkers are highly conserved among species. They're the most ancient of molecules. And so we find tremendous conservation from humans down to preclinical systems and even cellular systems in lower bacteria. Now, the challenge has always fundamentally been as to how we go about measuring these small molecule biomarkers. And this is where Sapient has made great technical advances in the development of what we refer to as high throughput mass spectrometry or next generation mass spectrometry. Now, these systems leverage what we call rapid liquid chromatography and couple it to high resolution mass spectrometry. Here's just a brief video of our actual laboratory that's based here in San Diego. Now, what these instruments fundamentally allow us to do is to take any biological specimen, whether it be plasma, serum, urine, CSF, tears, tissue samples, lymphatic fluids, normal tissue, all the way down from a clinical system to a preclinical system to cells in a dish. And in any of these biological systems, we can assay somewhere in the order of 11 to 15,000 small molecule biomarkers. Now, we do this very quickly. Our analytical cycle time for analysis of a given sample is approximately 53 seconds. And given the fact that we have a fully automated and robotic laboratory from freezer inventorying all the way through processing and mass spectrometry that's completely robotically controlled, this allows us to have enormous capacity. And today we can handle several thousand samples for analysis per day. Now, in each of these biological specimens, as mentioned, we can assay somewhere in the order of about 15,000 small molecule biomarkers. And if we look at these, approximately 1,000 of these markers can be identified readily up front using commercial standards, with the remaining unknown molecules being intensively classified with using what we call 4D chemical classification. And that imparts not only their retention time on a specific column, but also their accurate mass, MS1, their MS2 chemical fingerprint, and their CCS value on ion mobility mass spectrometry. This 4D chemical classification allows us to classify and label each one of these unknown molecules and ultimately isolate that molecule for identification as needed. Now, these high throughput systems are embedded in a very, very rigorous quality matrix, which allows us in real time to follow not only instrument performance, but chromatographic performance, column integrity, sample integrity, liquid handling, as well as many other parameters related to not only the technical aspects of our instrumentation, but also the integrity of the sample and the molecules that are being assayed. Now, when we think about what's known versus the full chemical breadth, these mass spectrometry approaches traditionally have really focused on that subset of molecules that are highly abundant in the body. And these represent only approximately 5% of the molecules that are present. By using the non-targeted discovery approaches here at Sapient, we're actually able to interrogate way beyond the 5% of known molecules as mentioned to really interrogate the full breadth of chemical diversity, which could span more than 75,000 molecules in blood in humans. Now, when we think about the types of chemistries that may be present, our technologies allow us to capture a broad measure of chemistries, everything from non-polar molecules all the way up to polar molecules. This includes both traditional metabolomics as well as lipidomics in a single run. When we think about those 800 to 1,000 molecules that are identified, these fall into a number of chemical classes, such as amino acids, nucleic acids, organic amines, central carbon metabolites, oxylipins and bile acids, as well as endocannabinoids, and many other signaling molecules, monoacyl phospholipids and nucleotides, triacylglycerols and cardiolipins, as well as tens of thousands, as mentioned, unknown molecules that may be of interest for which we perform 4D chemical classification and ultimately can isolate these molecules for structural elucidation. Now, we've coupled these technologies here at Sapient with a very large, what we call human biology database. Given the fact that our mass spectrometry systems allow us to perform bioanalytical analyses at an incredible rate, we've been able to go around the world and collect very, very unique biological specimens and analyze them through these mass spectrometers. These data, now that encompass over 100,000 human biosamples, have been amalgamated into our human biology database. This includes data not only from mass spectrometry measures, meaning these tens of thousands of small molecule biomarkers, as well as longitudinal clinical outcomes spanning more than a decade to now 3 decades of time in individuals. In addition, there's quite a bit of multidimensional other omics-based data, including genomics measures, protein and proteomics measures, microbiome data, as well as many other clinical features, including demographics and geography, response to various interventions, lifestyle factors, including diet, and as mentioned, adjudicated longitudinal clinical outcomes data over several decades of time. Now, really coupling these mass spectrometry data with this clinical information requires deep biocomputational analysis. And here at Sapient, we have a deep biocomputational team that has expertise not only in the handling of mass spectral data, but also multidimensional integration of these small molecule biomarkers with genomics and proteomics, as well as mentioned the clinical outcomes data, preclinical data, pharmacodynamic modeling, and many other factors. Fundamentally, this biocomputational approach allows us to take very complex multidimensional data and elucidate actionable single insights that we can rapidly translate forward. Now, here's one such example. We spend quite a bit of time thinking through multidimensional integration, including integrating genomics measures with small molecule biomarkers. Within the Sapient database, there's over 20,000 individuals in which we have dense genomic measures with corresponding blood metabolite measures through our mass spectrometry systems. And this allows us to fundamentally statistically begin to analyze this type of data. What's shown here on the bottom is a modified Manhattan plot looking across these 20,000 individuals with integration of the entire genome with over 50,000 circulating small molecule metabolites. Now, what we find from these integrations linking genetic and small molecule biomarkers is that over half of the metabolites circulating in blood have an actual genetic relationship, meaning we can actually map this molecule to a variant within the human genome. Now, if we look across all 20 million variants that are present within the human genome, just over 5% of these have an association with a circulating molecule. And importantly, if we look at those genetic variants that lie within the transcribed gene region, so they're actually coding within one of the 20,000 or so human genes within the human genome, we find that over 90% of these polymorphic variants actually have an association with a circulating small molecule biomarker. Now, this is an extremely important observation that allows us to do a number of things. For instance, we can begin to understand the biochemical origins of any single circulating biomarker through its integrating with genomics and understanding of those enzymatic processes and transporter systems that allow for that biomarker to be formed and transported into blood. We can also take the reverse approach and look at disease variants that have previously been implicated in a host of different diseases and ask, are there circulating markers that may read out particular genetic variants and allow us to understand the genetic architecture of human disease? In addition to these approaches, we can leverage these integration between genetics and small molecule biomarkers to discover new target engagement biomarkers that reveal actual genetic integration and interactions that may occur, again, from small molecule, large molecule, or gene and cell therapies, and develop readouts within the blood that tell us if genes have particularly been engaged and targets have been engaged in their actual tissue of origin. Now, fundamentally, this is a tool, again, that allows us to align patients, their disease processes, and specific therapies. And there's many particular applications that can be used to accelerate drug development, both in thinking through patient stratification and safety profiling, companion diagnostics and clinical trial enrichment, understanding disease processes and identifying new targets and target validation, thinking through early disease detection to disease progression biomarkers, as well as understanding therapies, both dosing, timing, target engagement, and pharmacodynamics, as well as the toxicology of specific therapies. Now, what I'm hoping we can do in the last 10 minutes or so here is to take a moment and to walk through some real-world data. And these are some case reports we're happy to share with you today that talk about how we actually may use these approaches for a variety of different clinical trial enrichment strategies, both for early diagnosis of a common disease, for diagnostic modalities in the rare disease population, and also thinking through their application in clinical trials, including in clinical trial failures. So this first example highlights the application of these approaches for understanding early disease diagnostics, which is obviously a very important area and is intimately coupled to drug development. This is a program here within Sapient that is focused on understanding non-alcoholic steatohepatitis, or NASH, which is an incredibly prevalent disease worldwide given the obesity epidemic. But this is a disease for which diagnosis has proven to be challenging, typically requiring very advanced MRI-based imaging and or biopsy of the liver, which is an extremely morbid and challenging procedure. And so we've been quite interested in being able to identify a circulating diagnostic marker that reveals patients with NASH at a very early stage in their disease process, precisely so these individuals can be enrolled for early clinical studies. And so this is a program that began with approximately 10,000 individuals in which we had advanced imaging of the liver, as well as liver biopsy in a number of folks, hepatic function tests, as well as adjudication of diagnosis of NASH over time. And when we analyze these 10,000 human samples that came from these individuals in this clinical study, we find, once again, there's somewhere in the order of about 15,000 markers that can be assayed in each individual. And given that what's in my blood may be a little bit different than any other individual across the entire population, there's somewhere in the order of about 50,000 total circulating small molecule biomarkers that are present. Now, we can simply begin by asking here on the left panel, in which each of these 50,000 or so small molecules is revealed as a gray dot, which markers are elevated in those individuals that go on to develop NASH relative to age, gender, and demographic matched counterparts. And when we do so, we find that there's a host of markers that are shown by the red dots on this left panel here that are altered in individuals that develop NASH after adjusting for the multiplicity of hypothesis testing here. Now, as I mentioned earlier, interrogation of these small molecule biomarkers to date has really been limited to a very small percentage of known molecules. And that's highlighted here in that when we look across all these thousands of unknown molecules, the most important molecules here for NASH are all unknown. So each of these red dots represents an unknown molecule whose structure has never been elucidated, and these have never been measured in any database in the world. Now, even in the absence of understanding the structure of these molecules, we can begin to organize them into pathways using what we call molecular networking, by which we can fragment these molecules and cluster them based upon their MS2 fragmentation patterns. When we do so, we find that 3 of these molecules here that are circled in blue on the left panel all lie in the same biochemical pathway. We can tell you how they're related to one another and what the differences are. These are unique markers that are all chemically very related to one another, again, lying in the same biochemical pathway. Now, on this middle panel, we can show this molecular networking. We can take one of these markers here that's labeled as the key biomarker. We can isolate out this marker. We can solve its structure. We can synthesize it for confirmation of its structure. It's solving an unknown molecule, and then fundamentally validate it in independent studies, both human studies as well as preclinical studies as shown on the right. Now, when we look at independent human studies in those individuals with now biopsy-proven NASH, what we find is with increasing stage of NASH, there's an increase in the median level of this single biomarker. Now, again, this is not a panel of biomarkers, but only a single circulating small molecule biomarker. Now, one of the lessons that we're continually retaught and learn over and over again is that even individuals, as I mentioned earlier, that share similar diagnosis and similar pathology on pathologic examination can have very different clinical outcomes. And in the case of here, if we look in all those individuals that all share a diagnosis of stage 3 NASH on the upper right, we find that there's actually two distinct populations. A population of individuals that have very high levels of this marker, as denoted by the pink box, and a population of individuals with stage 3 NASH that have a much more subtle elevation in this marker, as denoted by the pink and blue box. Now, all of these individuals would be enrolled in the same clinical trial, and we have reason to believe that those individuals that have a high level of this marker relative to a lower level actually are developing stage 3 NASH for very different biological reasons, and as such are likely to have very different response to specific therapies. And this is an example by which we can use this singular biomarker not only for diagnosis, but also stratification of individuals for clinical trials. Now, as mentioned earlier, these markers tend to be highly conserved, and when we look in a preclinical model, as shown on the bottom right here, we find that this is a marker that's elevated very early on in the disease process, as soon as that there's inflammation at the site of the liver, whereas if we look at individuals, or here preclinical animal models of a precursor state of NAFLD, we find that the marker is relatively flat. This allows us to study further the marker as well as its underlying biology. Now, as I alluded to earlier, one of the real powers of Sapient is our human biology database, and this allows us to take a discovery and amplify it over 100,000 individuals very quickly. And shown here is how we go about doing that within the context of this NASH marker. And so given the fact that we have 4D chemical characterization around this marker, even prior to underlying its structure, we can map this into our human chemical database and understand its relationship with both NASH as well as related phenotypes. So when we look across more than 100,000 people, as shown here on the left, given the fact that we have dense longitudinal information, the first question we can begin to ask is how early on in the disease process is this marker altered? And what we find is that people that have high levels of this marker relative to those individuals that have low levels of this NASH biomarker, again, this is a single circulating small molecule biomarker, irrespective of age, gender, geography, demographics, insulin, glucose levels, et cetera, what we find is that this marker denotes individuals who are going to go on to develop NASH at a much higher rate, up to 10 years in advance of actual formal diagnosis. So this is once again a marker that's released into blood at a very, very early stage in the disease process, prior to any clinical presentation or formal diagnosis. And this is a means by which we may be able to enroll these individuals for early clinical studies and early interventions prior to terminal tissue injury. In addition, we can begin to evaluate the dynamic nature of this marker, asking how does it change with age, with gender, with geography, and with time. We find that this is a marker that's remarkably stable over the course of the day and doesn't change with diet or exercise, fasting or feeding, circadian cycles. This is also a marker that's stable over longer periods of time, both a month, a year, as well as up to a decade in normal individuals. So it doesn't change based upon normal aging processes. It's a marker that's remarkably stable, once again, over geography and in individuals irrespective of diet and lifestyle. What we do find, however, is that those lifestyle interventions that result in improvement in NASH actually result in a dynamic change in this marker. So if we look in our human biology database and those individuals before and after bariatric surgery, what we find is that those individuals that have very high levels of this marker, indicative of severe NASH, after bariatric surgery have an appreciable drop in the marker, almost down to normal levels. And this is in correspondence to a change in actual liver inflammation and liver infiltration. This suggests that this marker is in fact a dynamic marker, and this has very important implications for how we may leverage this one biomarker as a surrogate endpoint in early clinical studies to evaluate potential efficacy of agents even prior to changes in ultimate pathology. Now, as mentioned earlier, also we can begin to do quite a bit of multidimensional integration that allows us to provide tremendous insight around any of these markers, including many of the thousands of unknown markers. And here's one such example by which we may integrate genomic and small molecule biomarkers. So if we take this one single NASH biomarker that's been discovered here in our human biology database using our genetics information, essentially we can do the genome-wide analysis around the circulating marker, and that's what's shown here in the modified Manhattan plot on the left. And in doing so, we can identify those genetic variants that occur in enzymatic processes and transporters that give rise to this marker. Given where these enzymes and transporters are expressed, we can actually confirm the tissue and cell type. And for the sake of this NASH biomarker, can confirm that this marker originates within the inflamed hepatocyte and even the organelle within the hepatocyte that the marker is being formed in, as well as the mechanisms by which it's transported from the intracellular state into the blood. We can also begin to evaluate causal inference using a variety of statistical tools, including Mendelian randomization. And when we look at those common genetic variants that influence the level of this biomarker on the x-axis and ask how does that map onto development of disease across large populations of over 500,000 individuals as marked by liver ALT on the y-axis, what we find is this exquisite linearity by which those genetic variables that increase the biomarker also increase risk of disease. And when we see this linear relationship, it's extremely suggestive of a causal relationship, suggesting that this biomarker, as well as the mechanisms that give rise to this biomarker, are causal drivers of NASH. And so in doing so, we can identify not only an early diagnostic marker that is quite dynamic that we've been able to validate in addition to preclinical systems, but we've also identified here unique targets now in NASH that may intervene upon as part of a therapeutic strategy. Now, this is one such example in common disease. We also spend quite a bit of time thinking through rare disease processes and how we can discover not only diagnostic markers, but markers that may be extended from the rare disease space into common disease areas. One of the areas that we've spent quite a bit of time thinking through is mitochondrial disease and identifying circulating markers that read out mitochondrial dysfunction. Now, this is a program that started in over 100 individuals with sequence-verified mitochondrial disease across a host of different mitochondrial pathways and over 1,000 of the proteins that are present within the mitochondria. And what we find, once again, when we analyze these biological specimens, measuring thousands of circulating small molecules, as shown here on the left, that there's a number of biomarkers that are elevated specifically in individuals with mitochondrial disease relative to those age, gender, and demographic matched counterparts. Now, you may be able to read on the y-axis of this left volcano plot that these are highly significant markers, approaching 10 to the minus 30, 10 to the minus 40 for association with mitochondrial disease relative to control counterparts, as shown in the small inset box. Now, once again, these are all unknown markers. When we begin to do pathway analysis, we find that all or a good portion of these markers, as denoted by the red dots, actually once again lie in the same biochemical pathway. This is a biochemical pathway that's never been described in humans and once again represents a new target for potential therapeutic intervention. We can isolate out one of these markers as shown out here by the blue dot, again, solving its underlying structure, developing targeted quantitative assays around this singular marker, and validating this marker in independent human studies. In an independent human study of mitochondrial disease, we find on formal ROC analysis that this one single biomarker has an AUC of 0.8, which is significantly greater than current state-of-the-art genetic sequencing, which is quite challenging in these individuals that have thousands of potential variants that may be pathogenic. As such, this is a marker that's been advanced now to our high complexity CLIA laboratory here at Sapient for development of a clinical assay that ultimately will be used for diagnosing these individuals. As we showed the example with NASH, we can also begin to use our human biology database to understand this marker better. Through formal GWAS analysis, again, we can identify the enzymatic processes as well as the transport systems and confirm, in fact, that this is a marker that's produced within the mitochondria and then is released by various transport systems into the blood where it's present, and we can assay it non-invasively. Now, within these rare disease populations, we spend quite a bit of time of thinking about how we may be able to leverage our human biology database to extend therapeutics that are developed in the rare disease space and apply them for common diseases. And as we're well aware, there's many shared biological processes between rare diseases and common diseases. Of course, in the case of mitochondrial dysfunction, this has been implicated in a host of more common diseases, including physiologic aging in individuals. When we look across our, again, database of over 100,000 individuals, irrespective of where one in the world lives or one's underlying biology, what we find is that this is a marker that linearly increases from mid-age. So somewhere around age 50, this is a marker that starts to elevate linearly throughout the course of one's life up until age 100. And again, this is analysis across more than 100,000 diverse individuals from around the world. This is some of the first real-world evidence that mitochondrial dysfunction, again, as marked by this single circulating biomarker, is altered with the process of chronologic aging. Now, when we look at individuals, irrespective of their chronologic age, we find that those individuals that have high levels of this marker, relative to age-matched individuals that have low levels of this marker, have a much worsening mortality over time. And that's what's shown here in the middle plot. Across 30 years, what we find is that individuals with high levels of the marker, again, indicative of worsening mitochondrial dysfunction for a given age, have almost a 25% increase in overall mortality, and suggesting that this is a marker not only that reads out a particular disease or a particular biological process, but may also be leveraged for understanding survival in individuals. Not only does this read out overall mortality, but this marker is associated with the development of specific disease processes, including both incident heart failure as well as type 2 diabetes, as shown on the forest plot on the right here. These are both diseases for which mitochondrial dysfunction has previously been implicated in preclinical systems now. And this single marker, once again, is identifying subpopulations of individuals with these diseases that are particularly being driven by mitochondrial dysfunction and for which interventions in this space may prove to be particularly useful. Now, the final example I want to share with you here is really around our clinical trial strategies and how we can use pharmacodynamic biomarkers to predict drug response. And this is an area that has been of particular high interest to Sapient and a number of our sponsors and clients with whom we work, particularly thinking through failed clinical studies and how we can once again begin to understand why a drug may have failed and how we can rescue these therapies by understanding specific biomarkers. On the left here is a clinical study that was performed together with one of our sponsors, in which we looked at a failed phase 3 trial of a rare disease in over 250 individuals, once again, in which a drug was failed to reach its primary endpoint. When we analyze the blood samples from these individuals, we find that there's a single circulating marker here that's circled in blue that was altered in response to development of or exposure to this particular therapy. Now, when we look on the top right here in those individuals that received this therapy, both at baseline as well as post-exposure, what we find is that for this single pharmacodynamic marker, there's a portion of individuals that have an elevation over time, as well as a portion of individuals that don't mount a particular pharmacodynamic response to this therapy and likely are not actually having effective target engagement. Now, when we substratify individuals based upon this single pharmacodynamic marker, once again, this was a failed trial, and we subset individuals based upon high versus low levels of this single biomarker. What you can see here on the Kaplan-Meier plot to the bottom right is that there's a great difference in that those individuals that actually mount a pharmacodynamic response to this given therapy and have real effective target engagement are protected from the disease, as shown by the blue line, whereas those individuals that do not mount an effective response to this therapy continue to decline precipitously, and as such, you can see a difference between these two mortality curves. This is an example by which we can now leverage a single pharmacodynamic marker to better stratify patients into potential responders versus non-responders, as well as potentially rescue an agent that had initially failed within phase 3 studies. Now, in the interest of time and to allow some time for some questions, I'm going to go ahead and stop there. I just want to thank the audience for their attention and for joining us today. We here at Sapient, as I mentioned earlier, are a discovery organization that provides discovery services to the biopharma industry. We'd be happy to chat with anyone who may be interested in leveraging these technologies and these data assets to enhance drug development. And I would encourage anyone to please reach out either by phone, by email at discover@sapient.bio, as well as through our Calendly link. Our website is www.sapient.bio. And once again, I look forward to speaking with anyone who may be interested in leveraging these tools for their drug development purposes. I'd be happy to pause here and take any questions.
Great. Thank you so much, Mo, for your presentation. So I'd now like to open the floor for discussion. As Mo mentioned, please remember to send any questions that you may have using the question panel on the platform. So to kickstart the Q&A, our first question, Mo, is can you comment on the role of small molecules biomarkers relative to protein biomarkers or genomic biomarkers?
Absolutely. It's really readily apparent how genomic biomarkers have transformed certain disease areas, particularly within the oncology space, and allowing to understand as well as diagnose these oncologic diseases, and then leveraging that information for targeted therapies. Now, the challenge is, again, is how do we begin to understand those diseases outside of the oncology space in which genomic markers have been more limited in their application. And this is where we very much view small molecule biomarkers as a complement to traditional genomic and or proteomic markers. Now, what makes these small molecule biomarkers very special, as I had mentioned earlier, is that they can be produced within the cell, again, whether it be normal to cells, whether it be diseased cells or the microenvironment cells, and they can be readily released into blood. Now, as we know, the vast majority of genomic markers, as well as proteomic markers, are retained within the cell and necessitate biopsy in order to interrogate these markers. And this, again, was one of the great differences with these small molecules that can be released into blood, and which we can non-invasively measure, as we've shown in several examples, as a means for complementing other genomic or proteomic markers. So these very much, again, are a complementary data modality that allow us to understand disease pathobiology and align patients with their disease processes and specific therapeutics.
Great, thanks, Mo. So our next question is, you're looking at tens of thousands of molecules that generates a massive amount of data. How do you go from R&D discovery to single biomarkers that can be advanced to actual clinical tests?
Absolutely, and this is a really good question, so I appreciate it. As you can imagine, rapid translation is a core component of what we do here. And we leverage these high throughput mass spectrometry systems in order to assay tens of thousands of markers in large populations quite quickly. Now, this is where we use our biocomputational and advanced statistical approaches to go from this large tsunami of data to single actionable insights. And as I showed you in these examples, this oftentimes involves measurement of only a single biomarker. Now, here at Sapient, we have a high complexity CLIA laboratory that allows us to very rapidly develop a CLIA-based assay around those single markers. And these are highly quantitative, targeted assays using traditional mass spectrometry systems that can thereby be deployed externally, either to other CROs or even internally within pharma organizations for advancing their drug development programs. And so there's a whole pipeline of translation that's embedded here at Sapient in our R&D efforts that allow us to go very quickly from initial R&D and screening through technical validation, biological validation using our human biology database, as well as ultimately development of these targeted clinical assays that can be deployed externally.
Great, thank you. So our next question is, how did you begin to link biomarkers associated with genetic variants causally to disease?
Yeah, it's a really good question, and an area again that we spend quite a bit of time thinking through together with our pharma sponsors. And as you can imagine, there's been an explosion in our understanding of the genetic architecture of disease, in which we've been able to identify a number of genetic variants that associate with disease processes. However, making sense of this information has been far slower and really challenging. And how we begin to develop drugs that target these specific genetic variables has been really quite slow over the last decade. And this is where these circulating biomarkers can be quite helpful. For one, through data integration, as we showed you an example there, and linking of genetic and small molecule biomarkers, we can begin to cluster gene variants that have shared pathobiology, and in doing so begin to understand the pathways by which these genetic variants may be influencing disease processes. We can also develop circulating small molecule biomarkers that denote or read out specific genetic variants their biology and their target engagement. And this has proven to be quite useful in the development of therapeutics, whether they again be small, large molecule therapeutics and/or gene or cell therapies targeting specific genetic variants. And so this is absolutely an area that we've spent time thinking through together with our pharma sponsors, and have a number of sort of wins, if you will, in which we've been able to leverage our data assets and these small molecule biomarkers in order to understand genetic variants and then leverage that information as part of the drug development pipeline.
Thank you. So we've just had a couple more come through. The next question is, how do you incorporate small molecule biomarkers practically into a drug development pipeline? And then what are some of the applications for the biomarkers you discover?
Absolutely. So as you can imagine, again, these are a tool that can be applied across the continuum of drug development, all the way from very, very early preclinical studies and identification of new targets and target validation, all the way through preclinical studies in toxicology, and then ultimately into the phase 1 through phase 4 clinical studies. Now, at different stages, we can begin to ask different questions of these markers. Again, in the early clinical studies, typically focused on target identification and validation, toxicology, pharmacodynamic biomarkers, and target engagement markers. And as we advance into the clinical studies, typically we can leverage these tools to begin to understand not only target engagement, but substratification and companion diagnostics that may denote individuals that respond or not respond to particular therapies. Now, how these may be applied varies again by the specific drug development program. For early studies, of course, as I mentioned, this can be used to identify new targets and de-risk these targets. In the latter stages, oftentimes we're using these approaches to understand how particular therapeutics may be operating in individuals and develop the next line of therapy or companion diagnostics that may be leveraged to better apply these therapeutics, both in successful as well as failed clinical studies.
Thanks, Mo. So the next question is, what opportunities do you see for mass spectrometry in supporting personalization of therapy?
Yeah, it's a really good question, and certainly is at the crux and the heart of what we do here at Sapient. You know, genomics, once again, and the introduction of parallel sequencing has been transformative for how we understand a number of disease processes. And to be able to go from where we are today to that next stage of truly personalized therapies, we have to be able to introduce additional markers that complement genetic markers. And this is where I view small molecule biomarkers as being particularly transformative and having large potential impact over time. I would suggest that these markers, together with traditional genetic markers, will be instrumental in the near future, not only for early diagnosis of disease, as we showed you in the case of NASH, but also fundamentally in identifying the personalized therapies for individuals in a way that, again, allows us to align individuals with their specific disease biology and then ultimately with specific therapies that are targeting that specific area of biology. These markers are a complement, once again, if you will, and already are being leveraged as part of clinical medicine. If we think about creatinine or triglycerides or glucose as diagnostic markers of a number of disease states, and now being able to measure tens of thousands of these allow us to quickly evolve, if you will, these biomarkers from an R&D perspective all the way through translation.
Thank you, Mo. So we have time for a couple more questions. The next one is, you said many of the molecules you capture are unknown. How do you go about identifying biomarkers of it?
Yeah, so you're absolutely correct in that the vast majority of markers that are circulating in our blood today are unknown and that they've never been mapped or structurally elucidated anywhere in the world. And while this can be challenging, this also represents the great opportunity for novel discovery. And being able to assay these molecules is critical, as we often find that the most important diagnostic or prognostic markers or companion biomarkers lie in these unknown state, as we showed you the example both in common disease as well as rare diseases. Now, the way we advance these, of course, is through what we call our 4D chemical characterization, in which we're able to classify the chemistry of these unknown markers, again, using their accurate mass, their retention time, their MS2 patterning, as well as their CCS. And that allows us to map these unknown molecules from any biological specimen into our human biology database and rapidly validate discoveries using orthogonal clinical measures, genomic measures, etc. Now, once that validation has been done, we can isolate out these markers and identify them using traditional chemical approaches, including cryoprobe NMR. Ultimately, we're able to synthesize these markers and then develop the targeted clinical assays. And this is really at the core, once again, of what Sapient does, and it's how we enable discovery. And it's fundamentally critical that we're able to isolate out and solve these unknown markers. And so this is an area we spend quite a bit of time in.
Great, thanks, Mo. So I think we'll just take 2 more questions. So the next one is, can you comment on circulating biomarkers for rare Mendelian mutations versus common genetic variants?
Yeah, absolutely. So in both cases, circulating biomarkers can be very important. Oftentimes we find there's a gradation of response, if you will, in that a rare Mendelian disease may have a huge change in a circulating marker, as we showed, for example, in the case of rare mitochondrial diseases. And when we look at more common genetic variants that may be targeting those same genes, we see a more subtle elevation in that marker. A classic example, of course, is LDH cholesterol with Mendelian forms of hypercholesterolemia versus more common genetic variants in the LDH receptor. LDL receptor. Now, in both cases, these small molecule biomarkers can be quite important. And this is where our genetics database here at Sapient that contains both rare coding variants as well as millions of common genetic variants can allow us to understand circulating biomarkers that may reveal both rare pathogenic mutations or, again, more common genetic variants. In both cases, we can leverage this information not only for target engagement and understanding the biology of circulating drugs, of certain mutations, but also leverage this information as part of development of therapeutics that target both common genetic processes as well as rare processes.
Great, thanks, Mo. So on to our last question. In your case examples, you reduced a large number of statistically significant metabolites to a single biomarker. What is the benefit of doing this versus using a panel of several metabolites?
Yeah, it's a good question, and this really comes down to practicality more than anything else. As mentioned, rapid translation is at the heart of what we do, and we want to ensure that our discoveries are able to see the clinic and advance forward drug programs very rapidly. Now, there are cases in which we'll do much more advanced multidimensional integration with machine learning or AI approaches that essentially develop patterns or complex data patterns that reveal out subpopulations. At the same time, it's very important that we're able to reduce data down to single measurements for which we can develop CLIA assays. Now, which approach we take is very much dependent upon the underlying question, and we have the ability internally here to do both. Again, very complex patterning using AI, as well as much more reductionist statistics that allow us to identify single markers. And in the cases we showed, these were really driven by a desire to rapidly translate the discovery, and as such, there was a focus on a single marker. Now, there are cases in which a panel of markers on the order of half a dozen or less may also prove to be quite important. Looking through substrate products, for instance, and a ratio of those is one such example. And so there are instances where we've extended from one marker to a handful of markers for diagnosis or prognosis or understanding drug processes. And certainly we're not limited by a number, but more so by practicality and what can be developed under CLIA sort of based protocols.
Great, thanks, Mo. I think that's about all the questions we have time for today. I would just like to thank you again, Mo, for your presentation and for answering the questions, and for everyone for coming. So if you'd like further information regarding Oxford Global's Biomarker series, please visit our website, which is oxfordglobal.co.uk/biomarkers. So all that remains to be said is thank you everyone again for coming, and I hope you enjoy the rest of your day. Thank you.
Thank you so much.